Hello, and welcome to You Can and Must Understand Synthetic Biology. I'm Andrew Hessel, and I'll be walking you through a presentation on this new and exciting technology. The title of this talk is actually taken from a very influential zine that came out in 1974 by Theodore Nelson called You Can and Must Understand Computers Now. And it was really a publication that got many people interested in computers and media. And of course, we know where that took us. Um, by way of introduction, I'm a microbiologist and a genetic scientist. My background is in genome mapping. I started doing genome mapping of bacteria in the late 1980s, early 1990s, really some of the first sequencing projects. And more recently, I work with a company called Autodesk. Autodesk is a company that makes software for design and engineering, visualization, and simulation. It's usually best known for its AutoCAD line of products, which are used for the development of buildings and other complex systems, cars, vehicles, etc. But recently, Autodesk has started to explore the world of biological design and molecular design. I'll tell a little bit more about that later in the presentation and how it loops in. I'm also faculty at something called Singularity University. If you're not familiar with Singularity University, it's based at NASA Research Park in Mountain View, California, in the Bay Area. And it's a startup university that was founded in 2009, really with a completely different vision for teaching students about technology. And it was recognized that by a number of people in the Bay Area that most schools were backwards looking and really weren't doing a good job at teaching people about really fast changing technologies like computers. And so the whole idea of being able to teach about exponential trends, something we're pretty familiar with in the world of electronics, because Moore's law and the idea that computing power doubles every 18 months or so is pretty much baked into the digital world. It was realized that we need to teach people about exponential trends because they're not intuitive. As Peter Diamandis, one of the founders of the university, likes to point out, if you take 30 linear steps, you know exactly where you are. You basically walk across a room. When you take 30 exponential steps, you've taken over 1 billion steps and you've walked around the earth multiple times. We're just not trained to think exponentially very easily. So we teach people how to be leaders in exponential technologies and how to apply exponential technologies in the world in a number of different areas, including water use, global health, energy, etc. My particular area is in bioinformatics and biotechnology. And here I like to point out to people that really it's one of the first exponential technologies that cells are effectively living computers, and our genomes are operating systems. And starting to build on that foundation, an understanding of how genetic technologies are starting to mature and be advanced. And this ties in with synthetic biology. But first, before I go into that, I just want to do a quick review of really what exponential technologies look like. And I'll use computing as an example because they're so familiar. This is a picture of the first transistor, and it was developed in 1957 in Bell Labs. It really looks like something that was hacked together in a basement out of old parts. But this is the foundation of probably one of the largest industries in the world today, digital technologies. And there's a whole history of these technologies being developed in Silicon Valley. It's a fascinating story. But ultimately, very quickly, we advanced beyond the transistor into semiconductor technologies, where the entire computer was manufactured on a chip. This was a powerful change in the economics of computers, and it really sparked a number of innovators. Famously, Steve Wozniak and Steve Jobs founded Apple Computer in a garage, which is pretty remarkable. People like Bill Gates and Paul Allen founded software companies, Microsoft. And very quickly, we progressed into these computers, which were once only accessible by elites in industry and academia and the military. These computers showing up in the home and being used by individuals. And of course, this caused a massive expansion of hardware, networks, software, etc., creativity, really, and the explosion of the web. Of course, Larry and Sergey came along to found Google because it was hard to navigate the web, and built a powerful enterprise around this. Mark Zuckerberg, a few years later, recognized that it's really not computers that want to connect, it's people that want to connect. Really powerful shift in the understanding around networking, and more recently, Jack Dorsey and Twitter, creating essentially a nervous system for these technologies. Now, this digital technology goes very deep. It's bidirectional. Today, we're actually digitizing the world. Just about every major city is being digitized by laser scanning, photography, satellite photography, etc. We're mapping the world in completely new ways. We're also learning how to map individuals at high resolution. This is something we do at Autodesk, and we're able to apply, make characters in movies that never existed before. This is modeling and simulation that life science only dreams about. But it's also moving down to a consumer level. This is scanned artwork from the Asian Art Museum in San Francisco, just done with an iPhone. Very quickly, it can be digitized on the cloud and turned into models that can be manipulated digitally, very easily, and actually turned back into reality with things like the MakerBot and other 3D printers. So we're getting a full iteration, a cycle here of being able to scan the world, be able to manipulate it digitally, and turn it back into reality. Through our computing technologies that have been evolving so quickly. But what I like to point out to people is that living things is really the first exponential technology, and the technology that is ubiquitous to this planet. All these computer and hardware tools that we've engineered are fantastic, but life is really the first exponential technology, and it's a complicated technology. I studied E. coli and Salmonella for many years as part of the genome projects, and I was always fascinated by the complexity of what we consider to be this simple organism. Ultimately, David Goodsell has a beautiful book on the machinery of life depicting this organism, and as you go through these illustrations and start diving into E. coli, one realizes it is massively complex. It is a self-assembling, self-reproducing system that's quite beautiful. And of course, we see this under the microscope with bacteria as it starts to divide from one cell and ultimately through truly exponential growth to produce billions of cells in 24 hours. Now, what I started realizing working at the intersection of computers and life science a long time ago, I was one of the first people doing essentially bioinformatics, was that there was a relationship that was appearing that in my world, where I could see the physical layers that we were creating with computers had analogs in life science. Components were really the individual proteins. These circuits that we were making, you could think of as basic metabolism. And if you could put enough circuits together, you really got pretty much in the entire cellular metabolism. And if this was encapsulated and operating right, you have a living cell. And of course, cells have their own architectures. tissues, and organs, and which make up us. So really, one can think of life, multicellular life, as really being a network of living computers. In the case of a human, hundreds of billions of these computers, and it's networked all the way down. It's essentially information processing all the way down. Today, it's quite intuitive because we're so used to having these networks, these computer networks around us, that people coming into life science and having life science explained to them through the architectures of computing, it becomes very, very straightforward and understandable to them. Now, if you take an E. coli bacterium and you step on it, you can release its genome, so to speak. This is a famous micrograph. This is about 4.5 billion megabits of genetic code, more than sufficient to encode a free living cell. When I first saw this picture, I saw this image, and I found it just burned into my brain, and I realized I want to understand how this information is organized. Now, that was really bioinformatics, and what we today look at is systems biology. And this is largely mediated through sequencing technologies and bioinformatics. Today, sequencing technologies are very powerful. This is a machine called the Ion Torrent. It is a desktop DNA sequencer. It's very fast. It's not too expensive. It can do about a human genome in a day, based on chip technologies. We've seen a massive increase in our ability to read or scan DNA extremely quickly. We've also started to write DNA. The tools are still are becoming digital, and this is really the foundation of synthetic biology, being able to do digital manipulation of DNA. And instead of traditionally using recombinant techniques to build our designs, effectively the equivalent of taking preformed words and scissors and glue and creating new programs, synthetic biology is rooted in less analog technologies. Here's the type of equipment one actually needs to do genetic engineering in the classical form. But what's happening, and really the foundation of synthetic biology, is that the actual printing of DNA can be done digitally. So now we have the ability to read DNA very quickly, to analyze it computationally, and now to actually print it using companies like DNA 2.0, GeneArt, IDT, and other companies that are specializing as essentially DNA printers. Now, if you haven't seen a DNA printer before, here's a unit that's quite advanced. This is an array synthesizer. A chip gets loaded into this device, and they have different densities of chips. The chips are electronically controlled, and they grow on these chips individual strands of DNA that are precise to the base pair. These are then assembled to create longer constructs, and these array synthesizers are allowing us to get to very, very low cost per base in DNA synthesis. But the take-home message here is that essentially DNA synthesis has become push button. And this is a remarkable change for anyone that's been in the biotech industry before. Really doing this type of manipulation through a computer, using just a computer, was unheard of a decade and a half ago. And now essentially anyone that has access to a credit card, a computer, and the internet, and some of these DNA synthesis technologies essentially has the power of a major biotechnology company, the foundational technologies anyway, at their disposal. What's quite interesting too is that the economics of of reading, of analyzing, and ultimately printing DNA are truly on an exponential, in fact, a super exponential curve. This is the first person to really start quantifying this was Rob Carlson, who's one of the early synthetic biologists. And this curve, this graph of the cost of DNA sequencing and synthesis over time is started to be called the Carlson curve, really the biological equivalent of Moore's law. And the blue line here represents the cost of sequencing, which has seen dramatic improvements, up to 5 times the pace of Moore's law over on a year-to-year basis. And the yellow line represents the cost of DNA synthesis, which has been falling exponentially and looks to fall super exponentially soon over the next few years. So really, the cost of doing this is getting cheaper each year, and it's getting faster as the technology is improved. We're still limited in terms of the overall size of the program that we can create, but there's improvements coming down the path on that. So, what does one do with push-button DNA? Well, one of the first examples that we started to see in the design space was that it could be used to do DNA origami. This is essentially using synthetic DNA staples to bend a larger fragment of DNA into different shapes. This is precise molecular scale control using synthetic DNA to manipulate matter at the nanoscale. I particularly like the happy face. I think this is, it made me happy to see that this is work that was published around 2005 and 2006 by Paul Rothman. It's been extended into 3 dimensions. What's fascinating about this work is that, again, DNA is just being used as a structural material, but the biophysics of the material are so well known that it can be designed on computer and turned into these shapes very, very reliably. And in fact, these shapes are starting to get functionalized in different ways now. There's been some tremendous work coming out of the Wyss Institute where groups have made things like cancer-fighting robots by functionalizing some of these shapes in new ways. This is a technology that's still in its infancy, but it's moving, again, very quickly because we have these computer-based design tools and a printer. But DNA, of course, is typically used for genetic engineering. And now that genetic engineering has gone digital, we've been creating a new community of programmers, so to speak. This is a picture of the students that have reached the finals in a program called IGEM, or International Genetically Engineered Machines, which is a program that was started at MIT in 2003 to train synthetic biologists, or essentially these young engineers that wanted to go out and do genetic engineering, but using standardized and modular parts and that are ultimately being printed and synthesized. So this is really biological programming. To date, about 12,000 students have been through the iGEM program. Many of them have, the earlier ones have received their PhDs and are now taking positions throughout the world, and they're in high demand. And their capabilities are quite remarkable. These were some students from Cambridge about 3 or 4 years ago that were engineering E. coli that would glow very, very brightly, with the idea that one day they would be able to make glow-in-the-dark trees. Wonderful technologies, but it's not just today. There's over 200 different institutions around the world that participate in the iGEM program. It's one of the main gateways into the field of synthetic biology. But there's also a large number of high school teams that are coming in as well. So this is a technology like computer programming that's getting pushed down to lower and lower levels because the tools are making it easy to access, and of course the costs are still falling. In fact, I started to see evidence of this back in 2008 with the formation of a group called Do-It-Yourself Biology, which essentially grew out of the iGEM community and students that said, you know, we love doing this so much, we don't really want to do it as a once-a-year competition. We want to make it part of our everyday lives, and starting to play with these things and learn about different aspects of life science, even if we're not going to be full-time in learning life science. And this organization has grown remarkably, again, exponentially over the years, and is now, there are thousands of people on the mail list that are actively engaged, and there's and many projects being done. I also started to see the appearance of people taking DIY bio into the real world in starting small companies. And this is a fellow by the name of Cathal Garvey in Ireland, a very active member of the DIY community that created a licensed biotech company in a spare bedroom in his mother's home. He actually does genetic design and sells his designs, and loves making equipment and hacking equipment, and a very staunch supporter of open technologies in life science. But it's moved beyond just individuals with a passion to groups working together in communities. And this has been, we've seen evidence of this with appearance of labs like this. This is a community lab in Brooklyn called GenSpace that uses essentially a gym membership as their business model. For about $100 a month, you could become a member of the lab, take various courses, and learn how to do various types of genetic engineering. There's projects across the range of life science in this, and they're certainly not the only lab any longer. There's a lab on the West Coast called BioCurious that's quite large. There's a new one that's just popped up in Harlem. And around the world, there are now dozens of these community biotech labs. So there's, just like we saw in computing, it's moved from specialized communities to highly trained individuals with a passion to share it and grow community, to essentially garage biotechnology, and now to more of a grassroots growing exponential community of developers. This is also starting to move into this activity is moved into the funding realm as well. I showed you the students at Cambridge that did the glowing, that wanted to make glowing trees one day. This is a group that decided they'd actually try and do it, or take the first step anyway. They started they put a project on Kickstarter, which is a crowdfunding site on the web, saying that they wanted to make glow-in-the-dark plants that ultimately had a luciferase gene, and they received 8,000 backers and almost $500,000 in funding. It's a little controversial because they plan to give out seeds as part of the rewards for their funders, but it's legal, and it's certainly changing the discussion around biotechnology and the release of genetically modified organisms, even if even if they're benign. But this is really all early markers and indicators of fundamental change coming into life science. We're also seeing that some of the students that came through the iGEM programs over the years, and they were actually training students, when they finished their doctoral work, they decided to not go into postdoctoral fellowships, but into industry, again with a very open ethos. And this is a picture from Ginkgo Bioworks, located in Boston. The founders are some of the original instructors of the iGEM program. Their goal is to unabashedly to genetically engineer organisms. This is a shot from their website. Their aim is to be able to do complex metabolic engineering in E. coli on contract and create new, essentially, new metabolic end products that have been tuned for various feedstocks and customers. Very ambitious. This type of engineering 10 years ago would have required another order of magnitude of investment and R&D activity. So this is really a powerful step. An area I've been watching very quickly because it's still expensive and slow to engineer a microbe is the development of viral engineering using these technologies. The first synthetic virus was booted up about in 2002 by Eckert Wimmer. He did the proof of concept. It was the first fully synthetic genome. He synthesized a poliovirus genome and demonstrated that it could be booted up and make active particles. That was very controversial at the time, but since then, dozens of viruses have been engineered using synthetic biology techniques and for applications including diagnostics, more recently vaccines, and I think very soon we'll start to see a number of new applications in therapies, cancer therapies, and eventually gene therapies as these get robust. Currently, today's DNA synthesis costs making a large, something like an adenovirus genome would cost about $7,000. Adenovirus having a genome of about 35,000 base pairs. But we can quickly see as these technologies fall in cost, it could be as little as under $100 in the next few years to do this type of engineering in a very rapid and high throughput way. So this is forcing a rethink of the application space and also how this type of industry should be monitored, regulated, etc. Quite a fascinating area that I've been very closely tracking, but has tremendous applications just because it can rapidly iterate. I'd like to point out that it's probably in some ways more amenable to regulation and monitoring than virology of days past because it's being done through a digital medium. So there's a lot of recording of the transactions. Probably the most sophisticated use of synthetic biology to date has been Craig Venter and Ham Smith at Synthetic Genomics in the J. Craig Venter Institute, who demonstrated 3 years ago now that they could synthesize and assemble a complete microbial genome. This made world headlines, but I think it's telling. This is about 1 million base pair genome. I think it's telling that this work has not yet been repeated even 3 years later to my knowledge. In fact, this is, as to my knowledge, the only synthetic bacterium that has been built from scratch. Last month in London, there was the 6th conference ever in synthetic biology, known as SB 6.0. The keynote speaker was a fellow by the name of Chris Voigt, who has been working to create high throughput ways to re-engineer not whole genomes, but sections of genomes that we call operons. And developing an architecture, a programming language, and ultimately the automation to do this. Chris has been working to re-engineer nitrogen fixation, the nitrogen fixation cluster from Klebsiella, and essentially port it to E. coli. It's a large block of DNA, about 20,000 base pairs. It's been refactored. The original control elements in the wild type genome have been stripped away, and very robust switches and other control mechanisms have been imported. The first time he did this, this actually lowered the fitness of that particular operon, but in his most recent work has been able to essentially re-engineer it up to wild type specifications again. This is really powerful tools for metabolic engineering in living creatures, and getting and not necessarily always understanding what nature created, but being able to apply engineering rules to make it more easy for humans to understand, manipulate, and ultimately optimize the systems. Moving beyond bacteria, which is still organisms that are unfamiliar to most people, there's baker's yeast. Yeast is, for the last several years, there's been a project underway to build a synthetic yeast genome from scratch. This is an ambitious project because the genome is much larger, the chromosomal structures are much more complicated. It's being pretty well funded now, and it's moving at a pretty good pace. It's in partnership with the United States and with BGI, the sequencing and synthesis powerhouse in China. What I find fascinating about the yeast engineering work is that people are very familiar with yeast because, of course, it's the foundation of making bread and beer. And beer is I like to think of as one of the top 3 most important liquids on the planet. Some believe beer has actually been responsible for most of human civilization. I'm not so sure about that, but what I do know is that it's getting accessible to bioengineers. This is an iGEM team from 2012 from Munich that engineered 4 different beers as part of their summer-long project. They introduced a protein-based sweetener, a protein-based flavor into the architecture of the beer, a cancer drug, and in a stroke of genius, the entire biosynthetic pathway for caffeine. What a group a few years ago at Rice University in 2008 had also engineered cancer-fighting beer. But what was remarkable at this group from Germany was they actually made the beers and filtered out the yeast, so it's not a genetically modified organism, and ultimately had some taste testings done with the beers as well. So this is, I think we're going to see a lot of engineered yeast moving forward, and potentially a new bio beer industry starting to emerge with some very different flavors and perhaps even therapeutic qualities to the beers, nutraceutical, so to speak, and very likely a few recreationals as well. So now we've covered basically that you've got DNA synthesis, allowing to do structural engineering at the nanoscale, cellular programming at the nanoscale, reprogramming, synthetic cell construction, synthetic virus construction. This is still being done pretty much at the single cell level. What happens when you want to go multicellular? Well, multicellular engineering is actually fairly difficult. One of the things I've been tracking very closely is the use of bioprinters to externalize the positioning of individual cells, essentially taking development into a much more human controllable process. This is work done by a company. One of the companies at the forefront of the space is called Origami that I've been tracking over the years. And Origami buys this 3D printer that is capable of printing cells. Their core technology is around cell engineering, and today they're successfully making synthetic liver tissues that they're able to sell to pharmaceutical companies for standardized toxicity testing, etc. I think as this technology gets more robust and cheaper, we'll see a lot of new applications in this. People are already talking about making synthetic leathers and synthetic meats as the cost drop so that we can essentially grow fewer cows. But after that point, there's even the possibility of using synthetic or muscle cells that are deposited by something like a 3D printer to also make for robot locomotion, very small robots and controllable as well. So some cyborg technologies. So these are powerful foundational tools, but some of the key that I want to point out here is it's basically digital design coupled with some sort of digital printing that can be controllable. This is really the foundation of a lot of synthetic biology. Another interesting application is really just backups. DNA turns out to be one of the most robust materials for digital storage. George Church recently demonstrated that it can be used as digital backups for computers. But we're also learning that we can go back in time and actually start seriously thinking about de-extincting some of the recently extinct animals. And we'll probably see this technology used as a way of preventing extinctions and enhancing conservation efforts as we move forward. But this is really the forward fringe of this technology. I like to point out that, in computers we say things get faster, cheaper, better, smaller. Certainly in the short term, these technologies and genetics are going to allow things to go faster, cheaper. I don't know if they'll necessarily be better, so I usually say faster, cheaper, weirder, because we're starting to see people explore very different spaces than could be explored by the conventional biotechnology industry or even academia in the past, because they've essentially, the costs have reached a toy or hobbyist level. I like to point out that it's really just going to be simplified to design on computer, print on some printer, and ultimately, if it's legal and safe to do so, shipped to the individual. Of course, this is prompting a rethink of biology and biological regulation pretty much at all levels. Mark Goodman, a cybersecurity expert, and I wrote a piece for The Atlantic last year discussing how we really need to start thinking about things a little differently, and it was called Hacking the President's DNA. And we meant to be provocative because we spend a fortune on biosecurity, but how is biosecurity changing in a world where these technologies are going digital? I don't necessarily think it's going to make things less safe in the short term. In fact, I think today the biggest risks in the digital world come from computer technologies. But we have if we look at life as another programming medium, I think we really need to start considering what some of the dynamics that we've seen appear in the digital world of computing are going to appear in life science. And are things like privacy issues, things like spamming issues, and hacking of various systems. So expect to see a lot of social dynamics in the field of synthetic biology as these technologies continue to advance and emerge very quickly. My own application focus, though, and my interest in this area is really rooted in the understanding, is in drug development. My background was with a major biopharmaceutical company, and after I'd spent several years with them, I realized we have a challenge in that the outputs of the pharmaceutical industry globally have been declining as measured by drugs per $1 billion invested in R&D. And this is a universal problem. It's not confined to one country or one company. It's really the global industry isn't a technology per se. It may use technology, but the outputs are actually appearing and taken to market not faster, better, cheaper, but actually slower, much more expensive to develop, and a lot of me too or very incremental improvements to the drugs. So this is, I believe that this trend has the potential to be reversed through core fundamental technologies like synthetic biology that change the economic paradigms, potentially to the point that we can actually consider making personalized medicines, I mean truly N of one medicines, which can then disrupt things like the regulatory and clinical trial paradigms. So I expect to see a lot of activity in the drug development area, particularly in with respect to rare diseases, end-stage cancers that aren't responding to, or cancers that aren't responding to conventional treatments, and other areas that are just confined to individuals. And fundamentally, on a more philosophical level, as you know, really this in some ways is the end of natural selection, because as these technologies get cheap and potentially synthetic genomes on the scale of a human or a plant or an animal could fall dramatically over the next decade or two, to the point where they cost a few dollars or pennies. I like to say that the bottom of DNA synthesis is essentially the cost of cell division of one of our own cells, which of course is trivial. All the machinery is already in our cells. So this could really represent the end of natural selection in a way, because human intention could be intrinsically linked to the generation or conservation, or in fact de-extincting of living creatures on our planet. So along with computing, this is a really fundamental technology that we're seeing changes. So just to summarize then, synthetic biology, you know, many people argue about the definitions, but I think this is a pretty good one from a recent report from the National Academies. Synthetic biology is an emerging discipline that combines both scientific and engineering approaches to the study and manipulation of biology. The scientific approach is really the research aspect, the reverse engineering of existing metabolism, and learning to apply it to the engineering and manipulation of biological systems. And I think this is moving very quickly, you know, because it's ultimately, at its most basic, it's really just genetic engineering, only empowered and done with digital tools. So it's nothing new, but the way we do it is completely new. And we've seen these shifts before in the past with things like word processing, other digital technologies. Already there is a global, diverse, and highly interconnected community in synthetic biology. They communicate using electronic systems. They are sharing data using electronic systems. So it's really built on the foundation of conventional IT. And I believe that it is potentially unpredictable, just as some of the new applications of digital technologies are still surprising to us today. And because of its unpredictable, it has the potential to be highly disruptive. I'll end with this statement, which is a quote from the director of BGI at a recent meeting. He says, you know, really moving forward, we can really, he offered the slogan for the field of synthetic biology as life is what we make it. And I think on that note, I'll start taking a look at some of the questions here. Let's see. I've got, let me just take a look here. And some of the questions that I've got, this one doesn't have a name. Let me see. Pardon me, I'm just getting used to this interface. This is from Anil at Manipal University. If we're able to rewrite our DNA, then will it be possible that one day humans will be resistant to all diseases? I think that's a fascinating idea. I think definitely as we're probably going to be one of the last creatures to completely rewrite our DNA because of the ethical concerns in that. But we're certainly going to start applying this to model organisms sooner than later. And who knows, as we get more proficient at doing this and confident in our abilities to rewrite DNA, we could very well start to do that for human genomes. And I would imagine that one of the things we would try and do is make ourselves resistant to some of the biggest diseases of the day. I like to think that I believe that when it comes to one of the big diseases is cancer, of course. I don't know if we'll necessarily be able to engineer that out of our genomes, because really we're talking about disk corruptions. But I think that it's very likely as we start to re-engineer genomes, we'll be able to remove some of the disease causing uh, uh, code. But I think that's really, I think we have a long way to go before we start doing that. But there's already evidence that that we'll start doing this with various other genomes. Uh, this next question is from William at the Inland Institute of Aesthetic Dentistry. Hi, William. William writes, he's concerned enough that responsible scientists are playing with DNA, let alone community clowns. This stuff is very dangerous and amenable to simple mistakes, to say nothing of terrorist potential. Um, this is definitely one of the biggest concerns about the field of synthetic biology, that scientists don't know what they're doing. There's the potential for biological errors, and also that this technology can be used to produce weaponized versions of different diseases, etc. It's an absolutely common concern. So people from the very beginning have been discussing and thinking about how to put in safeguards. Um, I like to point out that the number one safeguard that we have in all of this is that unlike computers, we actually have a universal defense system in most living organisms, and it's our immune systems. Even bacteria have forms of immune systems. All higher organisms have defensive systems of some type. So biology is always at war with other biology. Um, and certainly these technologies allow for the creation of new or novel bioweapons, but they will all be synthetic and ultimately created through the architecture of digital tools. I'm not saying that this won't lead to potentially some new bioweapons, but they won't be done in secret, though there will certainly be processes to go and hunt down those that are abusing it. There are already safeguards in place that are in the software and in the printers, and I think it's just really I think it's really important that the world stays engaged with the potential risks around this. That being said, these technologies right now are largely being done by scientists that are properly trained, that are properly that have the right cultures of responsibility. This is certainly appearing in the DIY community as well. There is a new defense community growing up around this that is emerging quickly. I think that that we have a lot to learn, but we're learning a lot of the dynamics from what we're seeing in electronic computing. And for anyone that thinks electronic computing is less benign than these technologies, just think about in how many systems today computers are embedded. So there's risks at all levels with any technology. But I truly believe that the benefits of doing synthetic biology outweigh the risks at this point, because we're still essentially working with toy technologies, and we're really being cautious as we move forward in being able to track individuals, communities, tools, dynamics, etc. But again, be engaged and teach people properly. The next question here is from Mun Chin Lee from CISA. And the question is, what is the response of big bio, corporations, government agencies, etc., towards the theme of openness, open source, open access, backed by the DIY community? That's a great question. Right now, big bio has, if anything, been engaged in actually funding some of the explorations of this space. and watching it very carefully. But I've seen some large groups go and actually fund the iGEM program. They're very interested in building an open architecture, and I think in some ways it's because they realize that they're getting uh, essentially they're seeing some of the changes in their own industries, how difficult it is to produce things. They see this technology as potentially allowing them to accelerate their business. We're seeing some of the synthetic biology companies that are appearing in this space also becoming very economic. Intrexon just 2 or 3 weeks ago went public. It is now worth $3 billion. We've had other companies like Amyris go public. The companies that are doing gene synthesis are doing very well. So I think in general, Big Bio is looking at this very carefully, and the open source and open access foundation, isn't completely new to them, in the sense that the digital architectures that almost every company uses today, really with the web, has an open foundation on which proprietary layers have been built. So I think at this point, everyone's learning about the new intellectual property ideas, what it means in terms of participants, what open source versus proprietary looks like, and we may even see the appearance of new business models. So again, stay tuned to see how this plays out. Certainly in computers, proprietary and open source, neither one is completely deletes the other. Let's see here. Gennaro from the University of Miami asked the question, what do you think of how gene networks will influence disease? Again, another great question. That comes more in the area of systems biology. In fact, we're still trying to figure out many of the gene networks and how they actually contribute to metabolism and disease. I think we're just going to see so much more effort done here, and success, because it's getting easier and faster to sequence genomes of all organisms. The computational tools are getting smarter for reverse engineering them and for running virtual experiments on the data, looking for patterns, etc. So I think as we start to sequence more and apply more computational resources and get better at doing the analysis, we'll figure out a lot of these gene networks and how they contribute to not only disease but to healthy states. I was just up in Canada last week participating in the Personal Genome Project Canada. I fully intend open sourcing my genome. I want to see millions of genomes flow into the R&D databases so that we can start to make more sense of this sort of thing. And of course, we're seeing the opening of the microbiomes as well. William from the Inland Institute of Aesthetic Dentistry, again, another comment. Also think that we should be spending more time on messenger RNA to control processes, less likely to permanently damage the genome. Yes, very good point. There's multiple layers of control in cells. Really, the work that we're doing today is largely on microbial systems and single cell systems, but we have to learn really how to manipulate all these architectures in robust and predictable ways if synthetic biology is truly going to thrive. Daniel Greenberg from Kobo Products, Inc. How do you see genetic engineering and synthetic biology in general taking the next step into becoming a larger industry? Most of these small biotech companies seem to be startups and limited in their growth. It's true. This is really a field that's growing bottom up. The big companies have largely looked at synthetic biology as just a tool, whereas some of the smaller companies coming into the space look at it as a foundational technology and are driving it hard. About 2 years ago, I started to notice a shift in that some of the VCs and backers were starting to fund smaller companies in this space, and the money was really starting to flow in bottom up. It wasn't just going to the big name scientists, but starting to go to smaller groups. And I think we'll see that trend. Certainly in computing and software development, we see things like Y Combinator that fund hundreds of small startup application companies because the infrastructure for computing exists. So really, it's a very low-cost process to boot up a lot of companies. I think we'll start to see that appear in life science over the next few years as the infrastructure for synthetic biology and the use of things like fabs to boot up these technologies, virtualize it more. I think we'll start to see a lot more money flow into the space, both at the top with big industry making investments, but also at the bottom from VCs coming into smaller groups. Wow, there's a few more questions. I won't be able to get through them all, but Hector from Karolinska Institute. On the one hand, if I'm not mistaken, DNA origami only uses DNA as a raw block material. On the other hand, what do you think about DNA? And I can't see about DNA programming concerned with implementation of Boolean gates. So I won't go into this in detail, but Hector, you're right. It's absolutely, with DNA origami, it's used as a structural material. We are seeing many applications of DNA computing, essentially using some tricks with DNA to do some calculations. We've also seen just a few months ago from Stanford the instantiation of the first biological transistors with really tight junctions. So we are truly seeing computation come to these living systems, Boolean computation come to living systems and living and biomaterials in new ways. And I think that's very exciting. We'll see a lot more explorations. Probably have time for one or two more questions here. Martin from the Masaryk University. What do you think is the biggest biological obstacle, not the technical one, on the way to synthetic organisms? And do you think we should worry about extant organisms being replaced with synthetic organisms? I think the biggest biological obstacle today is still the core technologies of DNA synthesis are still very at the beginning. It's still very expensive to write a bit of DNA. We're not proficient at doing complex circuits in this work yet. And so we're not just manipulating single proteins like the first generation biotech industry, but we're still learning how to make more complex circuits. So I think we have a long path to go. I don't worry so much about extant organisms in the short term being replaced with synthetic ones, but I do think we are going to start seeing more and more synthetic organisms appear, first in controlled environments and later with ultimately getting licensed for release. But I think it's a very long process. What I do know is that, because we can have ownership of these things, it is an industry, people will make investments, and I see the emergence of boutique biotech and biotech, genetically modified organisms that people seek out because they have features that they want and are willing to pay for. But it'll be a long process yet. And probably the last question here from Ezra at the Istanbul Technical University. I think there's a dilemma between synthetic biology and natural life, because I believe if our society will get away from the natural organisms and processes, consequences of it will be disaster. Synthetic biology field seems perfect, but sorry, I just have to read. But have you ever thought about its impact to nature? Absolutely, and I don't have all the answers for this. What I really believe is that we'll find the right balance as we move forward as a species. I think it's a technology that's very powerful in its potentials, that we're really starting off, and some of our worst case scenario fears are probably shaping our ideas on this right now. But like computers, I see genetic engineering and synthetic organisms having large and generally positive impacts on the world moving forward. And when we see negative impacts, I think we'll learn from them very quickly. And perhaps this last question from Ruba at the Arab International University: can this technology help us increase human lifespan? Can it cure diseases like diabetes and cancer? And I think absolutely these technologies will have a big impact in cancer in the short term and with things like diabetes. cancer in particular, because I look at that as a disease of N of 1. But in terms of lifespan, we've been learning how to live, healthier, and that's allowing us to live longer. I don't think we're going to be engineering ourselves anytime soon for longer life, but we're every step of the way we're learning with our own use of technology. We're learning how to apply it to live longer, healthier, more fulfilling lives. So I think this is just another tool in our technology arsenal, but one that is that has very profound and far-reaching implications. Thank you for listening, and I hope you continue to explore the field. Thank you very much.